So this project was funded by the IMI initiative called Im2Pact, and it's a big European consortium, and it was really fantastic to be a part of. So I was on that project as a postdoc, and it was kind of split up into different areas. So the idea of the whole consortium was to better understand what changes occur in the blood-brain barrier across different types of neurological diseases, including MS as well as Alzheimer's. And then my part in this project was to better understand how the blood-brain barrier is affected in obesity and type 2 diabetes. And my arm of that work was using in vivo models. So I work primarily with mice. And to do this study, we used two different types of mouse models. So we used what we call a diet-induced obesity model. So this is using a standard mouse known as a C57, and we put it onto a high-fat diet for about 16 weeks from the age of 6 weeks old. And the second model we use is an obese mouse model, but it's also severely hyperglycemic as well. So it's a little bit closer to a model of uncontrolled type 2 diabetes, and it's more commonly known as the DBDB mouse. And with these mice, we did a lot of physiology with them, metabolic phenotyping. But one of the really exciting things we did was some brain blood flow imaging, and we also did some single cell sequencing. So that's where the project kind of started and where it grew from. So like I say, we had these two mouse models. The sort of distinction between them is one is diet induced and is just really obese, and it might be mildly hyperglycemic. And the other mouse model, it's actually very difficult to get any mice to be hyperglycemic. It's a lot it's a lot easier to get humans to be hyperglycemic, as we see with type 2 diabetes. But what this mouse has is actually a leptin-receptor mutation in, and what this allows it to do is become hyperglycemic. So those two mouse models, that's what we were working with, and we thought it was really important to look at functional outputs. So to do this, we did some brain blood flow imaging using a Moore laser, which is a laser speckle, but it also has the ability not just to look at the blood flow, but also to look at the oxygenation levels of that blood as well. So it's a pretty cool bit of kit. And the nice thing about working with it is it can do through skull brain imaging. So we can actually, all you have to do is sort of bring back the skin, but you can actually image straight through the skull. And that allows the brain to stay the right temperature as well as pressure as well. And in our study, what we found is that the high-fat diet animals actually had a reduction in the baseline blood flow. And then we also use stimulus to see if you can stimulate a blood flow response. But what we saw there is even though they had this reduction in brain blood flow, when we gave that stimulus, there was actually no overall difference compared to their controls. However, when we looked in our obese with hyperglycemic mouse model, what we actually saw was not only a reduction in the brain blood flow, but we actually also saw a reduction in the brain blood flow and a reduction when we gave a stimulus as well. So it's a lot more complex situation when that hyperglycemia is added in. And on top of that, we actually saw a reduction in the oxygenated and deoxygenated levels of the blood as well. So what we were seeing is quite a strong functional output of that obesity with hyperglycemia compared with just our obese animals. We actually did a technique called single cell sequencing. So what happens here is you can take the brain and you can isolate out cells of interest. So we're interested in the blood-brain barrier cells. So what we did is we isolated the microvessels, so the small blood vessels from the brain. And then we enriched for the main cell type that we were interested in, which is endothelial cells. So these, if you look at a brain blood vessel, most of the cells there will be endothelial cells. And these are really important cells because they control a lot of the transportation of oxygen and nutrients across the blood-brain barrier from the blood into the brain. So very important. And as I mentioned before, we did a technique called single cell sequencing. So what this allows us to do is look at the individual genetic profiles of these cells. And that's so powerful because prior to single cell sequencing, what we were really looking at is sort of groups of cells, and we weren't really able to differentiate the genetic changes in in individual cell types. So this allowed us to look at these genetic profiles really specifically, and we're starting to unpick a lot of the changes that are maybe happening that is leading to this functional changes that we see. And this data is still ongoing, so there's a lot of emerging pathways coming out there, but one of the key pathways that actually saw, so when we did what's called a KEGG pathway analysis with all our genes that were either being upregulated or downregulated, one of the main pathways that came out actually was Alzheimer's disease. And then I do a lot of sort of bioinformatics, and one of the things I like to do is what's called a gene screen, or what I call it is a gene screen, where you can pick any sort of genes of interest that you want to see how they're being changed in these conditions. And what we found in one of those gene screens for genes that are linked to Alzheimer's disease is actually that there was an upregulation of a gene called APP, which stands for amyloid precursor protein. And for anyone familiar with Alzheimer's disease, they'll know that amyloid precursor protein sort of starts at the beginning of the cascade that leads to amyloid plaques, which is a pathological hallmark of Alzheimer's disease. So what we saw in our data was an increase in expression as a result of obesity and hyperglycemia of APP in endothelial cells. So this is something that we will be following up on because we think it's very, very interesting. The Alzheimer's Research UK conferences are fantastic. I highly recommend them to anyone who's working in that field. And it's a really friendly community and people really want to support and collaborate. So many discussions after a lot sort of discussions around about neurovascular couplings. That's basically, you know, when the neurons signal with the sort of vasculature and blood flow changes. And also a lot of discussions on the data set itself, because it's absolutely massive. And we do have a lot of immune cells in there as well. So that would be really interesting to follow up and sort of look at some of the immune cells and how they're behaving in the brain as well. So yeah, lots of discussions and lots of really good questions as well.